Hey everybody, in the last video we took a look at enhancers, right? Enhancers in eukaryotes and how they influence transcription of target genes. And we looked at a specific enhancer in humans called KLK3E and probably other mammals. And what we saw was that the KLK3 enhancer is transcribed by RNA polymerase II to make enhancer RNAs. And these enhancer RNAs are required for the function of the enhancer. In this video, I am going to describe a really neat technique called Chromatin Conformation capture, or another name is just a 3C assay. 3C standing for chromatin conformation capture. And I want to start just by going over some of the key steps in this technique, and then I'll show you how it works. Essentially, what it allows a researcher to do, it allows a researcher to detect interactions between distant DNA loci. So it's really neat or really useful for detecting interactions between enhancers and target genes at distant locations in the genome, distant locations from the enhancer. So what are the key steps? So essentially we need to start with some cells. Then we need to treat the cells with a chemical called formaldehyde. And what formaldehyde will do, it will cross-link or form cross-links between the various chromatin inside the nucleus. So let's see. Cross-links between, I'll just say DNA loci. That sounds better. So it will form covalent cross-links between various loci that are close to each other, and it will lock those into place, keep them from separating from each other when we should finally extract the DNA. So the next step is going to be to permeabilize the cell membrane and nuclear envelope. So the cells are already dead, right? We treated them with formaldehyde that will kill the cells and lock the DNA into position. So we don't need to worry about permeabilization killing the cells. They're already dead. But we need to, permeabilize them. That's a difficult word to say, huh? So we'll need to do that because we need a restriction endonuclease. So we're going way back, I think, to the first lecture. So restriction endonuclease, we need to add one to the cells to digest the DNA. Then, and remember, restriction endonucleases, we're going to use one that targets a specific site. And it might be like EcoR1, which only cuts, GAATTC. So that's an example, restriction endonuclease that we could use. Then we are going to isolate the DNA and add DNA ligase. And that's going to join some of the fragments together. It seems kind of funny how we're digesting the DNA and then isolating the DNA and adding ligase. But I'm going to show you how that can allow us to detect interactions between distant loci. So step 6, what we are going to do, we are going to reverse the formaldehyde crosslinks. And we can do that by heating the reactions at like 65 degrees for a long time. Sometimes some of these protocols go overnight. And so that will, when I say reverse the cross-links, it will essentially break the cross-links between the DNA molecules. Then we will perform PCR to detect interacting loci. Now, there are ways to use this technique with high throughput sequencing, and you can identify all the different interactions between all the different loci in a cell. The version I am going to describe to you is when we have a hypothesis that predicts an interaction between just a few loci, and we are using this assay to test our hypothesis for just an interaction between just a few loci. So, and we're going to look at this technique in the context of a study that I touched on in the previous video. So, how do you say this name again? I think it's Assay. Oh, Shay. I think some people say Shay. Some people say, the pronunciation will be difficult for me, so I'll say Shay for now. Shay et al. I believe it's a Chinese name, but I'm not 100% confident on that. So, but this is a really amazing study. And what these authors were doing was they were looking at, the KLK3 enhancer. So here is the KLK3 enhancer. And essentially, they showed that the enhancer RNAs from this enhancer are required for its function. But they also use the 3C assay to show interactions between the enhancer and the promoters of a distant gene, a distantly located gene. So here's a crude diagram of the enhancer and some genes they are studying. So here is the KLK3 gene. Here is the KLK2 gene. Again, this is the KLK3 enhancer. I wonder if I can color these in quickly without wasting too much time, because I have some of these diagrams already drawn below. And to keep everything consistent, I'm going to try and color some of these in. Okay, that works. I usually use that color for the enhancer and red for the promoter regions. Okay, so the red are the promoter regions of the genes, and the light blue is the enhancer. Now, the authors had a hypothesis, right? So they think that eRNAs are important, and they think that eRNAs, the enhancer RNAs, are important for, helping a certain chromatin structure, 3D structure, form in the cell that allows the enhancer, or that brings the enhancer in close proximity to the KLK2 promoter. So it would look something like this. So that's going to be the enhancer. Here is the KLK3 gene. Here is a big chromatin loop. Here is the promoter of KLK2. Here's the KLK2, and the DNA continues off the page there. So I think I can color these in quickly again. And hopefully you're taking notes as you're watching the video. You can use some colored pencils also. So, okay, so their hypothesis is that the KLK3 enhancer is brought into close proximity of the KLK2 promoter. And okay, so when is it brought into proximity of the KLK2 promoter? So there is a protein called the androgen receptor that I'm going to diagram like this right here. So, okay, what is it called again? Let's say it is called the androgen receptor. And this is a transcriptional activator that will bind to the KLK3 enhancer when a molecule called dihydrotestosterone, DHT, binds to the androgen receptor. When that happens, androgen receptor will go in to the nucleus and bind to the KLK3 enhancer. So, and the authors have a hypothesis that when this happens, okay, so that's what induces or helps promote transcription of the KLK3E enhancer RNAs. And also, in the enhancer RNAs might be important, or probably important, for recruiting what's known as the mediator complex, which is a co-activator. That is not going to work. Okay, let me color that in. Okay, so this is the mediator complex, giant complex of approximately 30 protein subunits. Generically, mediator is referred to as a co-activator because it works with the transcriptional activators to activate transcription of target genes. In this case, the transcriptional activator is androgen receptor. It's binding to the enhancer, and mediator is helping promote transcription of the KLK2 target gene. And here's KLK3 up here. Okay, there are a number of things I haven't diagrammed. I haven't diagrammed the general transcription factors. I haven't diagrammed RNA polymerase II on either the enhancer or the KLK2 gene. But we have all the details in place to see what the hypothesis is. So the hypothesis is the KLK3 enhancer is brought into close proximity of the KLK2 promoter, and bringing KLK3E in close proximity to the KLK2 promoter requires enhancer RNAs. So the authors are going to use the 3C assay, the Chromatin Conformation assay, to test this hypothesis. So, okay, how are they going to use that? So hopefully in your notes you have the list of steps I went over. And one of those steps was, get some cells, right? Okay. Now they're going to have some cells that have been treated with DHT. So androgen receptor can go into, can bind to DHT and then go into the nucleus and bind to the enhancer and help rearrange the chromatin in a way that allows for transcription of KLK2. And then they're going to have some cells that they don't treat with DHT, and those are going to be the control cells. But beyond that, after all those steps, what they're going to need to do is choose a restriction enzyme, and I believe they use an enzyme called BSTY1. So BSTY1 is going to cut DNA throughout the genome at wherever there is a BSTY1 site. And I believe, you don't have to remember this, but I will say it looks something like this. This is RGATCY. That would be the restriction enzyme recognition sequence. I believe it looks something like that. And R stands for purine, and Y stands for pyrimidine. And it's going to cut DNA throughout the genome in those fixed cells, those cells that have been treated with formaldehyde, wherever there is one of these sites. Now, in this diagram down here, that is similar to the diagram I had before, where we have KLK3E here, KLK3 gene here, KLK2 gene here. The orange lines, that's where I'm saying there are BSTY1 recognition sequences. So BSTY1 is going to cut at all of these places. And the authors already know this because they have the sequence of the human genome. So they know exactly where it's going to cut. Now, once we know where we have a diagram like this and we know where BSTY is going to cut, we can define the certain fragments that we want to test for interaction in the 3C assay. So we need to define one fragment as the anchor. So the anchor is going to be way over here, right next to the KLK3 enhancer. And then we're going to test for interactions between the anchor and many other fragments at this location. So the authors looked for at this sort of approximate region like this. And so they defined fragments like A, B, C, D. So you can see this is fragment A right here. Between these two sites, this is fragment B, fragment C, so on and so forth. So we have fragments from A all the way to I. So we're going to use the 3C assay to see if this location over here interacts with any of these locations over here. More precisely, we're going to use the 3C assay to see which of these fragments represents the locus that interacts with the anchor most frequently. So we're going to identify which of these fragments the anchor interacts with most frequently with the 3C assay. But we're not going to do it, right? The researchers did this. We're going to see how they did it. So back to the hypothesis, right? So I have this model diagrammed again, showing the KLK2 or KLK3 enhancer in close proximity to the KLK2 gene promoter right here. Now I have all these little orange lines representing where the BSTY1 restriction enzyme is going to cut. And these pink lines, these are the hypothetical formaldehyde crosslinks that were formed between the various chromatin segments when the cells were treated with formaldehyde. Now, this is a conceptual diagram, so we'd have to, imagine that these DNA fragments are squished together, right? So when formaldehyde cross-links two segments of DNA together, they aren't separated by a large distance like this. These would be kind of squished together. So what I'm showing you is that these, with these pink lines, I'm showing to imagine that wherever the pink lines connect DNA, that those are sort of squished together in the cell. And they're squished together because the DNA has been folded in a certain way that has brought different segments of this segment of the chromatin close together, or this segment of the chromosome close together. So, and now we have our fragments defined. So the anchor is located way up here, and all the other segments are located down here. So we're looking for interactions between the anchor and A, B, C, D, so on and so forth. We are not looking for interactions over here. We would need to define other segments if we wanted to test for interactions between the anchor and any other location in the genome, such as the locations represented by the segments between the orange lines, say towards this end of the loop. So I forgot to mention that these red things right here, these are primers. So PCR primers. So each segment and the anchor need one PCR primer, and that's going to be important for detecting the frequency of interactions between the anchor and each of these fragments. So once we've done, let's see if I can clean this. Let me see if I can clean this up a bit. So once we have treated the cells with formaldehyde, permeabilized the membranes, added the restriction enzyme, digested the DNA. Now what we need to do is isolate that DNA. And so when we isolate the DNA, we're going to end up with pieces of the genome in a solution. And most of the pieces of the genome, if they're located far apart, they're going to, move away or float away from each other. But let's say we have this fragment right here. Let's say this is, the hypothesis is correct, and the KLK3 enhancer is brought into close proximity of the promoter of KLK2. If that is true, then we would expect a fragment like this to be floating around in that DNA solution. There's DNA from all over the genome, and various segments are cross-linked to other segments. But if the hypothesis is true, we should see this. We should see something like this in the DNA solution. Well, we won't be able to see it, but it'll be there. Now, once we have our DNA and we add ligase, so we're going to add ligase. So the ligase is going to join some of these ends together, right? So the ends are all compatible because we digested the DNA with one restriction enzyme. That's going to leave sticky ends, and those sticky ends are compatible, so all these ends can be ligated together. Now, what did I want to do with this diagram here? Okay, yes. I think I wanted to, okay, so we're interested in the anchor here, right? We're interested in identifying the segment A through I that interacts most frequently with the anchor. So when we add the ligase, let's look at this piece, this end of the anchor right here. We only need to look at one of the ends. And so when we add the ligase, where do we think the ligase is going to ligate that end? Or which other DNA end do we think the ligase is going to sort of stick to that end of the anchor? So we can probably, make it easier to see by just erasing all the DNA ends that are, too far away, because those are less likely to be ligated to the anchor. So here's a BSTY end there. There's one here. There's one there. One thing I want to point out is what I did is I just copied this thing right here, and then I erased. Everywhere there was an orange line, I just erased, because that's what I'm trying to do there, show you that the enzyme digested the DNA there and left a break. But the formaldehyde crosslinks are holding these segments close to one another. So when we add the ligase, well, the ligase is going to start joining the ends together because they're all compatible, but it's going to join the ends that are together depending on how close those ends are to one another in the solution. So if we focus in on the anchor, any end that is too far away from the anchor is less likely to be connected to the anchor than an end that is very close to the anchor. So let me erase, say, this piece right here, because we'll say, oh, that's too far away. So that segment is not likely to be connected to the anchor. And, okay, so neither is this one. It's kind of too far. I just want to go down to like 1 or 2 or 3 ends, the 3 closest ends. So the ends represented by I here. So that end here and this end here, well, those are too far relative to the others. So I want to erase those. Now we have segments G and H, and then we have this other one up here, which wasn't part of our assay. But theoretically, this end here could be ligated to here, could be ligated here, could be ligated here, or even these, which are a little bit further away. So I'm going to ignore these because they're too far away and just focus in on the most likely ends that the anchor will be ligated to. I'm going to call it, say, 1, 2, and 3. So 3 ends that the anchor will most likely be ligated to. So I have it diagrammed over here again. So let's say the anchor was ligated to this piece, this blue piece up here. That could happen. The anchor could be ligated to this end of the G fragment right there. And the anchor could be ligated to this end of the H fragment. And remember, others are possible, but the others are located. The other ends are fragments are farther away, so they're less likely than any of these. Now, after we add the ligase, we have to treat the DNA solution with high temperatures, usually 65 degrees for a long time, and that will break the formaldehyde crosslinks. And then what we could have is something like this. We could have a segment like this with the anchor ligated to that undefined fragment that we weren't really looking for an interaction with. We could also have the anchor ligated to segment G, or we could have the anchor ligated to segment H. And remember, we're analyzing millions of cells. So we could identify all of these different fragments or fused fragments could exist in our DNA solution. The one that should be most frequent is the one that represents the most common 3D structure of, or I should say 3D conformation of the KLK2 enhancer and KLK2 gene in the cell. I hope that made sense. If not, you'll probably see you're going to understand in a second. So, okay, for example, what about this one right here? This is segment B. Now we could go up here. Okay, where was B? Okay, B was way down here. Now, we're looking for an interaction with B, but that wasn't really our hypothesis, right? Our hypothesis suggested the KLK3 enhancer will be close to, F or G or E up here, not so much B. So B is a good control. If we see this a segment of, if we see B interacting with the anchor, then our hypothesis is wrong, and we would have to come up with a new one. A new explanation for the 3D conformation of the KLK3 enhancer relative to its target genes. So, okay, back to our original map showing the various BSTY1 sites in the various segments we're looking for. Now, depending on how the anchor is organized, the anchor segment is organized relative to these other locations, we could get all of these fragments. So what have I diagrammed here? Essentially what I did was I took all of these separately and diagrammed what the ligated fragment would look like if A was ligated to the anchor, if B was ligated to the anchor. And remember, A can be ligated to the anchor if the anchor is found close to that location inside the cell. And so here we have the various possible segments in our solution of DNA after we break the formaldehyde crosslinks. So we have A to anchor, B to anchor, C to anchor, D, E, F, G, H, and I to anchor. All of these are possible. So what we need to do now is use qPCR, quantitative PCR, with our primers. One for the anchor and one primer for each of the different segments. So the anchor primer is going to be the same to test the presence of all of these fragments. The primer, the fragment-specific primer, is unique to each fragment. So what we do is we use qPCR and see, which of these segments are found at the highest levels in our DNA solution, our solution of DNA. So what we're going to do is we're going to check the levels of each fragment by qPCR. So in this chart over here, what I have diagrammed, let me zoom out a bit, is just a crude chart showing relative levels of PCR product. On the x-axis, what A is, we're checking for interactions of the anchor with the A fragment. So we're checking for this fragment up here, A to anchor. B, we're checking for B to anchor. That means the qPCR involved the primer that was specific for the B fragment and the anchor. And we're testing for all of those fragments here. So, and what I've diagrammed down here is the result that the authors found when they looked at the cells that were not treated with DHT. So the androgen receptor did not go to the nucleus. It did not bind to the enhancer, and we wouldn't expect any rearrangement of the chromatin in response to binding of the androgen receptor to the enhancer, because there was no DHT. And so this is sort of the control, just looking for the basal level interactions between the various segments, A through I, with the anchor. And we can see, oh, there looks like, okay, the level of, let's say, E to anchor over here is slightly higher than the level of, let's say, A to anchor up here, right? This is slightly higher than this. Let me clean this up a bit. Okay, so when they do the same experiment with the DHT treated cells, I can use a different color here, what they see is essentially the same thing for all of the loci, let's say, except for these right here. Oh, okay, so what does that tell us? That tells us when cells were treated with DHT, DHT binds to the androgen receptor. The androgen receptor goes to the nucleus and binds to the KLK3 enhancer, and that brings the anchor fragment right here in close proximity to the promoter. And that's why we're seeing the anchor close to segments E and F. E and F right there. So really cool experiment, huh? I think so. I think this is amazing, where they add, a compound, and all of a sudden, in their after the 3C assay, in their suspension of DNA, they have a lot more of this fragment right here, the F to the anchor fusion, and this one right here. And it makes sense. Okay, that it seems like it must be because the cell organized the DNA in such a way that it brought the anchor, which is next to the enhancer, really close to the promoter for the KLK2 gene. Now, we're almost done. They do one more cool experiment, or two more cool experiments. Okay, oh yeah, I forgot I diagrammed this for you. Yep, okay, so those results are suggesting when the cells are treated with DHT that, okay, the cell brings this location over here close to the enhancer over close to the KLK2 gene promoter. And we have diagram down here, okay, the androgen receptor is bound to the enhancer. We have mediator mediating the interactions between the enhancer and the promoter. And we have the KLK2 enhancer close to KLK3 enhancer, close to the KLK2 promoter. And now we have evidence. We have evidence from the 3C assay showing that, yes, this indeed does happen in the cell when the cell is exposed to DHT. So now they did another experiment. So instead of doing something like this with all the different segments, they already know the fusion of the E to anchor fused DNA molecule. The levels of it go up when you expose cells to DHT, dihydrotestosterone. So they're just going to look at levels of the E to anchor segment in the 3C assay in response to different treatments. So we have high levels on the y-axis. We're going from low levels to high levels. And we're looking at 4 different cell cultures. One of them, or 2 of them, have no DHT. So we're just going to put that, okay, the levels of E to anchor fragment are relatively low when there's no DHT added to the cells. So what they're also looking at is they're going to use siRNAs, and we saw in the previous video, and I'm going to go into detail on siRNAs in the next lecture. But essentially what these do, for now, they destroy complementary RNAs. So if you have an siRNA, which is 21 to 24 nucleotides long, the cell will use that to look for a complementary RNA. No matter if it's a messenger RNA or an enhancer RNA, it will use the siRNA to find that RNA and degrade it. So the control siRNAs don't match anything. And what they found was that when, okay, they treat cells with DHT and they add siRNAs that don't silence or don't destroy any RNAs in the cell, that the levels of the E to anchor segment are high, just like they saw before. Because that makes sense. That makes sense. Why does that make sense? Because this is happening right here. And say this is the anchor fragment right here. And let's say this is segment E down here. Okay, so those levels go up because they've already shown that. But they looked at something new. Okay, they did something new. When they added siRNAs that matched the enhancer RNA from the KLK3 enhancer, I should use the same color. What they saw is that, whoa, levels were way down. The enhancer didn't work nearly as well as it did when those siRNAs for the KLK3 enhancer RNA were absent. So what this is telling us is that the eRNAs are important for bringing the KLK3 enhancer in close proximity to its target promoter. Now, what about mediator? So they did the same experiment for mediator. Again, when there's no DHT added to the cells, nothing really happens. There's no real rearrangement of the chromatin around the KLK2 gene promoter. When they use the control siRNAs that don't really match anything, okay, they see high levels of the E to anchor PCR product because the anchor has been brought into close proximity to the E locus, the E fragment, which represents that locus that is near the KLK2 promoter. Now, when they use when they add siRNAs that match the messenger RNA for a subunit of the mediator complex, the protein subunit is called MED1. When they add siRNAs that cause that MED1 messenger RNA to be destroyed, well, then the mediator complex doesn't work. And as a result, they didn't get nearly as significant nearly as significant an increase in the, bringing the KLK3 enhancer in close proximity to the KLK2 gene promoter. So what this is telling us is that the mediator complex is important. So we've got 2 things that are key. I'm sure there's many, many more things that are important for this process. But when it comes to bringing distant loci together, an enhancer and a target gene promoter, in this particular case, the enhancer RNAs are important and required for the process, and the mediator complex is required for the process. So in summary, 3C assays, chromatin conformation capture assays, detect interactions between distant DNA loci, or 3C assays can be used to detect interactions between distant DNA loci. In this case, we were looking specifically at the KLK3 enhancer and the KLK2 gene promoter. Also, transcriptional activation by enhancers. I'm going to qualify this because this is, relatively new work. We don't know if all enhancers work this way, so I'll qualify it by saying sometimes. But transcriptional activation by enhancers sometimes involves chromatin looping, the mediator complex, and enhancer RNAs. Okay, I hope you like that. We've been teaching this sort of looping model with enhancers for a while. If you took genetics with me, we learned about that, and now you actually know the techniques used to gather the evidence that something like this, some sort of chromatin looping structure, is important for transcriptional activation of target genes. Okay, I hope you enjoyed the video. I hope you really understand 3C assays now. In the next video, we are going to take a look at siRNAs, RNA interference, and eventually we're going to take a look at a relatively new discovery involving circular RNAs. And before we can look at that work, we need to learn more about siRNAs and RNA interference. Okay, so I hope you watch the next video and enjoy your weekend.